Hey, welcome to the first ever screencast for AP Biology. There's actually going to be two screencasts in order to cover this information. The first part will be on the cell cycle and mitosis, and then the second video will be on control of the cell cycle. Divide it into two chunks just to make it a little bit more manageable. Right now, if you could write down what you see on your screen, these are the things that we will be learning today. The first bullet point about how we're going to explain the cell cycle and mitosis. This is actually basically the same thing that you did in ninth grade honors biology, and even very similar to what you learned in seventh grade life science. The difference is that we will be able to explain why the different things happen in the events of the cell cycle, and especially knowing why this is so important. The next three bullet points are about how the cell controls the cell cycle. This is brand new information for this year. We will describe the role of checkpoints and how something called a cyclin and cyclin-dependent kinase will control the cell cycle. Last, we'll get to how the effects or what the effects of disruptions to the cell cycle might be. Most importantly, this is what can lead to cancer. Cell division, there are two types of cell division that have entirely different purposes. Please note that these are not opposites of each other. A cell doesn't choose which one to do. Instead, your cells will go through mitosis in order to create genetically identical diploid cells for growth or repair or asexual reproduction for unicellular organisms. These are your body cells or somatic cells. If you remember that mitosis makes your toes, that will help you to remember what mitosis does. However, meiosis makes me. This is going to be the process that we make genetically unique gametes for sexual reproduction. We will get to this topic much later. The cell cycle consists of three different parts. You'll notice this graphic below. While it's not totally to scale, it does show how interphase takes up more time of the cell's life than M phase by itself. So G1 is when the cell is growing larger because it has just gone through mitosis. It's also going to prepare for DNA synthesis. During DNA replication, this is the S phase or DNA synthesis, the DNA will be replicated. This has to happen in order to make identical cells. If you don't replicate your DNA, you can't split it in half because you only have one copy of it. G2 of interphase is when the cell continues to grow and, of course, do all of its normal cell stuff, and then the cell is going to prepare for mitosis. The M phase, this is mitosis. This is when the nucleus will divide to make two identical copies. This consists of prophase, metaphase, anaphase, and telophase. At the end of mitosis, we'll also have cytokinesis. This is when the cell itself divides to form two new cells. So if the cell were to go through mitosis but not cytokinesis, we would have a cell with multiple nuclei. This is actually what your muscle cells look like. Okay, so interphase, just went through the three parts on the previous slide, but we have growth, G1, the S phase, that is synthesis of DNA, and then G2, preparation for mitosis. The picture you see here are some actual cells looking at the different phases. Most of these cells are in interphase. We have one in telophase right here, a late anaphase, a slightly earlier anaphase. But the rest of these with just a dark dot for a nucleus, these are all in interphase. So even if these were located in a rapidly dividing area of your body, such as hair follicles or skin cells, or on the tip of a root, you would still see most of the cells in interphase. And here's a short little animation from Amoeba Sisters to help us understand the parts of the cell cycle. Mitosis always seems like a bigger deal but it's actually a very short part of the cell cycle. During mitosis, the DNA will condense to form chromosomes, and at this point, most of the DNA is not accessible by the cell. So while mitosis is essential to form new cells, the cell can't actually do a lot of its normal things because the DNA is all wound up in these chromosomes. Okay, so mitosis will definitely occur after DNA replication. If the DNA is not replicated for some reason, mitosis will not happen. The basic idea is that we're going to have sister chromatids that will be split to form two identical nuclei. As a short review, if we have a chromosome, we always draw it as an X. And what this X is showing is that we have two identical strands of DNA. Each part of this X is the same as the other one, and each part is called a sister chromatid, referring to the strands of DNA. And this animation helps us to understand what actually is going on during mitosis. Please remember it is a dynamic process. There's not a definite beginning and end to each phase. Instead, what we're doing is we're looking at how all the chromosomes are moving around. So, prophase, metaphase, anaphase, and then telophase. Okay, so the actual process of how mitosis occurs. The first thing is that the cell has to prepare for division. There are several different events. The replicated DNA condenses to form visible chromosomes. This is important because the DNA has to separate equally into each new cell. If we didn't have these nice, neat chromosomes, it would be very difficult for the cell to ensure each new cell got the same amount of chromosomes. Think about trying to separate Christmas lights. If they're all bundled together, it's really hard to get one strand separate than another. If you bundle each strand separately, it makes it a lot easier. Another thing that occurs is the nucleus and the nucleolus will break apart. This is so the chromosomes can move around without being tied up in the nucleus. We also have a mitotic spindle that forms. This will move the chromosomes around in the cell. They don't just float. They're going to be moved, pulled and pushed by the spindle. We also have centrosomes with centrioles located in them that will begin to migrate to opposite ends. Next is alignment. During the alignment part, the chromosomes are going to line up in the middle of the cell. To look at my little diagram over here, these green lines indicate the spindle fibers that are attached to the centriole on one end. Its function is just to organize the spindle fibers here. And on the other end, it's attached to the centromere of the chromosome. By attaching to the centromere, the spindle fibers are able to shorten to pull the chromosomes to each side, also to arrange them in the middle to begin with. Now, what is most important here is we need to understand why the chromosomes line up in the middle. If we can imagine an imaginary line going across the middle, well, now we can see we have equal number of chromosomes on each side. By lining up in the middle, it allows the cell to evenly split its chromosomes to each side. Okay, and then separation. The spindle fibers will separate chromosomes into the sister chromatids, and these chromatids are pulled to the opposite end of the cell. Now, in a human cell, right this very minute, how many chromosomes do you think there are? If you said 92, you are correct. Because remember that this is a chromosome with two chromatids. As it goes through the cell cycle, this chromosome is going to be split into the sister chromatids. Each one of these chromatids is the entire strand of DNA that contains all the genetic information. In other words, it's a chromosome. So a chromosome can be made up of two chromatids, and this one that has been replicated, or it can be single. Think about if you have copies of a sheet of paper. Each one has all of the same information. It's just whether they're in a stack or if they're separate. Okay, also during this separation, basically the cell is resetting. So the nucleus and the nucleolus reform, chromosomes uncondense. Um, and in a human cell, once we form our two new nuclei, now we're going to have 46 chromosomes in each of these nuclei of the new cells. Okay, a short little thing about cytokinesis. First of all, cytokinesis is division of the cytoplasm. So these two sets of cells show that we have two new nuclei formed. So mitosis has occurred. Now the cell itself is dividing. The top one is an animal cell. The cell will pinch in, and this place where it pinches in is called a cleavage furrow. And then as there's spindle fiber things that pull these together, we'll separate into two new cells. Now, on the plant cell, since it has a cell wall, it can't pinch in. Instead, we're going to form a cell plate in the middle. We have vesicles that fuse, and then eventually we're going to have an entirely new cell wall that's formed, and this will split the cell into two. Now, here's some pictures that we could see under a microscope in our classroom, or we can look it up right here, a little bit easier to look at. This is the end of a root cell. Pretty sure I got this from an onion cell to just show how each of these little dark stained things are where the chromosomes take up the color. So they're the nuclei. And we can kind of see where we have different phases of mitosis, but still glancing through there, you see that most of them are still in interphase. When you see some distinguished chromosomes, like in this one, that one would be going through mitosis. Okay, and then a zoomed up picture to show you what mitosis actually looks like in a real cell. We have interphase, and then we're beginning into prophase. The chromosomes are beginning to condense and continue, until we get to late prophase. And each one of those lines, those are the chromosomes. In an actual cell, it's not a nice, neat X, because we're talking about molecules that are not like stiff rods, they're more like string. Okay, metaphase, chromosomes line up in the middle. Anaphase, the chromosomes split. Remember, there are spindle fibers that are pulling them to opposite ends. And then we get into early telophase. Kind of tell there's a cell plate that's forming right here. And then we have telophase and then cytokinesis. We end up with two new cells with identical nuclei in each cell. Okay, that's all for right now. We'll come back to control the cell cycle in the next screencast.